our next speaker. Well, speaking of which, it's Dr. Katie Hinde. And without giving you too many of the details, I'm certain in a few minutes you'll know exactly why I'm about to rave about her work and her as a person. But in the mere, what would it be, Katie, 5 or 7 years you've been doing most of your work, Katie has elevated discussions and discourse and knowledge about milk beyond probably any segment of time at which we've learned something about it, and has really taken the direction, a unique and utterly fascinating direction of how milk itself organizes the expression of behavior, as she will be so beautifully telling you. She got her undergraduate degree at the University of Washington and went on to UCLA, got her PhD there in 2008. And Katie also has an important connection to the Comparative Lactation Laboratory. That's where you work at Harvard. And let's see here, she's a research affiliate of the Brain, Mind, and Behavior Unit at the California National Primate Research Center, the Nutrition Laboratory at the Smithsonian National Zoological Park, and the Foods for Health Institute at UC Davis. So I think one more thing. I wanted to make sure that you know she has an incredible blog. It's called Mammals Suck Milk, and I highly recommend it. Katie?
Can you guys hear me? Yes. Okay. Thank you for that incredibly kind introduction, Jim, and thank you, Darshan and Augustine, for inviting me to speak to you. I was incredibly flattered and excited for this opportunity because I think that this is a really important opportunity to interface about all the things that contribute to the building of babies, right? Be they non-human primate or human. And nobody in this room needs to know, of course, how important postnatal programming is, but it's worthwhile, I think, to revisit that most work on developmental programming addresses conditions during the period of conception or pregnancy. And this is because the earlier a nutritional insult or environmental experience occurs, the greater the magnitude of the phenotypic consequence. Okay? But for those of us who are interested in infants, the postnatal period is critically important. And this is because this is when the infant becomes behaviorally active with other individuals in a contingent manner. And so, of course, the first social relationship of a mammalian infant is with its mother, right? And 2 years ago, you had Steve Sumi here to speak, and I imagine, somebody correct me if I'm wrong, that he talked about this very stylized, contingent, communicative interaction between very newborn rhesus monkeys and their mothers, where they sit and they look back and forth and lip smack to one another. And it's very reminiscent of what we see with new mothers, with their young infants, and these contingent, communicative, facial expression, vocalization patterns. Okay? This is also a period where infants begin to explore their environment, both the physical environment and their social environment, right? And they learn and encounter all these new things. And this is also the time that they're gaining a wider degree of social experience, which is a very fancy term, I think, for saying playing, right? Because young infants and toddlers are doing a substantial amount of their activity in play behaviors with other individuals. Right? And these are very sensitive periods of neurodevelopment. Okay? And something that I think we generally take for granted is that all of this behavioral activity is made energetically possible through mother's milk, okay? From an evolutionary perspective, right? And so I will also spend much of this lecture convincing you that milk is not just about nutrition and has any number of other important bioactive factors that can be contributing to the behavioral development of mammalian young. So over the course of the talk, I'm going to familiarize you with mother's milk. I'll put it within the context of life history theory. I'll talk explicitly about how milk may contribute or seemingly contributes to behavioral development. And I'll talk very briefly about some fitness outcomes that we're starting to see as a result of this early behavioral programming. So the ability to synthesize milk from mammary glands is the defining characteristic of the mammalian class, right? It's where we get our name. However, synthesizing milk is one dimension of a broader lactation strategy. Part of the lactation strategy is going to be the duration that milk is provided from birth until the end of the weaning process, right? And so here at the top, we have the grand lactation champion of the orangutan, which seemingly lactates 5, 6, maybe possibly even up to 7 years. And at the bottom, we have the hooded seal, which lactates for a total of 4 days before weaning the infant. I'll return to why this might be, that there's so much variation. There's a frequency of nursing bouts. Okay, so this is the amount of time between suckling periods and the duration of the infant's suckling behavior. And so here we have the baboon female. She's locomoting, and the infant is on nipple while she's going about her day, right? And at least in the early neonatal period. And then you have rabbits, which only nurse once a day. So the mother is out and about and returns to the den just once to allow milk letdown and the kids to suckle. You have the number of offspring that a mother's providing milk for at a single time point, okay, so litter size. And in primates, this is typically 1 individual. But in other mammals, it can be up to like 22, for example, here in the tenric, which is this interesting little Madagascan rodent-like mammal. And within the size of the litter, and I'm not going to talk about it substantially today, but you can also consider the sex ratio of the individual offspring, so how many males to females. And then last, we have the milk she's synthesizing. Right? And we can imagine that the length of time lactation lasts, the frequency of nursing, the number of offspring being supported are all going to influence the amount of milk that a mother produces and what the composition of that milk is. And this is something that we tend to take for granted. All of us can go to the store and we can buy milk products. Right? However, when you think about it from a scientific perspective, our relative knowledge about lactation and milk synthesis is actually incredibly deficient. Okay? So here I've shown, this is just a basic bar graph using keywords inserted into PubMed, right? And this is the number of articles times 1,000. So when you put in pregnancy, you can see that the lion's share of research effort into maternal investment or infant development is actually being directed toward pregnancy. Much less so is lactation, or mammary, or breast milk, or milk composition, or what's really important, it's not just what the milk consists of. It's not just how much milk you're making. It's the two of those together that really tell you what milk is. Okay? And research in that area, as you can see, is particularly deficient. Most of the knowledge that we have about mother's milk comes from 3 areas. The first, of course, is dairy science. And this is very important for understanding the capacity of the mammary gland to synthesize milk. But from an evolutionary perspective, trying to understand how milk contributes to the rearing of an infant is very difficult in dairy animals because their milk rears us, right? Not their own calves. We know a lot about mammary gland function from cancer research. But obviously what I mean is we know a lot about mammary gland dysfunction. Okay? So that research is not looking at the mammary gland in its quote-unquote evolutionary natural state, in cyclical periods of lactation and involution. And then there is a body of literature that explicitly contrasts formula feeding to breastfeeding. And similarly, this is very limiting because it completely overlooks the natural variation among breastfeeding mothers. It doesn't tell us about differences within breastfeeding mothers about their milk composition. And it turns out that milk actually varies substantially among mothers in pretty much every dimension that's ever been investigated. So what is in milk? So it obviously includes all of the nutrients that we need for energy, right? So fat, protein, and sugars. Includes essential vitamins that you need to get from your diet, vitamin A. Has minerals. It very importantly has hormones. Right? And I'll spend a significant portion of today's lecture talking about cortisol in milk. Right? Cortisol is not in infant formula, okay? But it is present in human breast milk. We have immunofactors, so secretory IgA is an example, and we know that the immunofactors in milk reflect the mother's antibodies and are specific to her and her infant at the time of lactation. And we have to keep in mind that also water is provided to the infant through milk, so it maintains infant hydration. What determines the composition of a species' milk is very complicated. So you have things like the environment are going to be reflected in milk, right? So if you're in a very Arctic region, you need a lot of energy for thermoregulation for the pup or infant. If you're in an arid area, you need a lot of water in milk to support the hydration of the infant. Okay? And I told you about the hooded seal, lactates for 4 days. It transfers, its milk is 60% fat, basically butter. Mothers transfer 7 kilograms of fat per day of lactation to their seal pups, okay, because they have to put down a blubber layer in order to survive in this environment. And they do it for only 4 days because they give birth on these ice floes that are incredibly unstable and will disappear quite quickly. And they do this to avoid their primary terrestrial predator, the polar bear. So when we think about milk, we have to think very broadly about what are the environmental and ecosystem challenges that are facing mothers and infants during this period of infancy and lactation. Your milk is going to reflect what you eat, right? It's going to reflect the developmental priorities of the infant. This is important because what infants are doing during their development is sustained by the milk their mothers are providing. So how fast a species' infants grow is going to be very important for their milk. Faster growth requires more protein. Right? Slower growth requires more sugar so that you can maintain. So here I mentioned the orangutan that has a very long period of lactation. And this is thought to be because the diet that orangutans exploit is very difficult, and it takes a long time in order for infants to be proficient, right, at foraging for the food they need. So it's remembering where the food is, how to find it, and then a lot of the fruits and seeds they eat actually require 4 or 5 steps in order to access the nutrients inside the husk. Okay? Those steps have to be done in a particular order, and it takes a long time for orangutans to be nutritionally independent. Okay? And mother's milk supports them during this extensive period of learning. I have one quick example here of kangaroos. So kangaroos are born quite early. They're functionally embryos when they're born, and they attach permanently to a nipple inside the pouch. They spend a period of time growing, which you can see the joey's legs right there. And then they have this period where they can be in and out of the pouch and accessing milk. And what you find is the composition of the milk, sugar is very high when they're permanently attached, but that plummets down once they spend time outside the pouch. So here's a prime example of where the synthesis of milk and its nutritional composition specifically flips depending on the activity patterns and the developmental stage of the joey at that time. And of course, from an evolutionary perspective, the milk a species synthesizes is going to reflect their phylogeny. Right? Of course, to complicate matters significantly, these are all interrelated with one another. Okay? And so an individual species signature of milk is going to reflect numerous evolutionary and environmental factors. Okay, here is a phylogeny of all the primate species for which we have milk data on the composition. And you obviously don't need to know the species, but the important thing is that it represents all major clades within primates. And what you will be, I think, maybe surprised, or I'm not sure, excited to know, is that human milk does not look different than the milk of other primates. Okay? In terms of, along the bottom here, we have the percent fat, then we have the percent protein, and then we have the percent sugar. Okay? Here we have strepsirrhines, New World monkeys, Old World monkeys, apes, and humans. And in the gross composition of the macronutrients of their milk, humans do not look radically different from the other primates. And this reflects our slow life history. We have infants that grow slowly, they learn complex skills, and they have this extended period of infancy relative to other mammalian groups. But that's between species. what about within species? Right? Mothers are not all the same. And because they're not all the same, there's the possibility, and we now find to be the case, the probability, that their milk will vary among mothers. I'm particularly interested in life history theory. And life history theory tells us unequivocally that natural selection has not shaped adaptive traits in mothers to optimize their investment in an infant. Okay? Mothers are not shaped to maximize investment in an infant. Okay? Important take-home message. Natural selection has shaped mothers to optimize investment in a series of infants over a reproductive career. Okay? And so mothers have to balance investment in their current offspring against their opportunities to invest in offspring in the future. And we know this systematically. So you can imagine that depending on where a female's at in her adult reproductive career, she has different resources available for reproduction, and she has a different degree of horizon on her reproductive career. Okay? So you have young mothers that are initiating their reproductive career, and they have to allocate their resources to their own body maintenance, their own continued growth and development because they initiate reproduction before they're fully adult, and allocate resources to reproduction. And they also have the potential for a long reproductive career ahead of them. So they have fewer resources, more things to spend it on, and they have to protect their condition for future reproduction. So we can expect young mothers to show different patterns of reproductive investment than older mothers. You have prime age mothers who are no longer developing, so they only have to allocate resources to themselves staying alive and reproduction, but they have a shorter horizon on their reproductive career. And then you have very aged mothers who, and humans obviously live long after they stop reproducing, but in terms of the horizon of their reproductive career, it's catching up, and so they don't need to conserve their condition to maximize future reproduction to the same extent as prime and younger females do. Also, infants are not all the same. Right? They're going to vary in any number of parameters. One of the ones I've been most interested in is understanding differences in infant sex. Right? So, which I've anthropomorphized males here with a black bow tie. I don't know if you can see that out there, and females with a pink bow. The reproductive potential of sons and daughters is going to be different. Okay? And the social relationship of mothers with their sons and daughters, depending on the species, is going to be different. Okay? So in the macaque species I work with, males immigrate away in adolescence, subadulthood, sometimes as adults, but their daughters stay in the social group and form lifelong bonds with their mothers. Okay? And so we can expect that there might be differences in the milk synthesized depending on the sex of the infant. So let's actually look at some data about that. I do my research in, quote unquote, the monkey, which is the rhesus macaque, and it's the #1 biomedical model in the United States in non-human primates. Their genome has been mapped, which is very useful if you want to understand things about milk synthesis genes. And in their natural environment, much like humans, rhesus macaques are able to exploit a variety of habitats and ecosystems. Right? So you can find them in swamplands, rainforests, in high alpine areas, and in drier woodland grassland areas. And they also are incredibly successful at exploiting human-modified habitats. Okay? So they're what we call a weed species, like cockroaches, like rodents, like humans. The particular animals I work with are at the California National Primate Research Center in Davis, California. They live in half-acre outdoor corrals that are comprised of close familial kin, more distant kin, cousins, second cousins, and non-kin. This picture I calculated right here represents about 1/8 of their enclosure size. Okay? So they're able to exhibit a full suite of normative affiliative behaviors and agonistic behaviors. Right? And in this environment, they do not develop any of the behavioral stereotypies that can sometimes plague singly housed indoor non-human primates. Okay? Okay? And here's an aerial view because we work with animals living in multiple different corrals. Okay? So we're able to look at both within and between group differences. It's important to remember that rhesus macaques are seasonal breeders. So they breed in the fall in our population. They have about almost 6-month pregnancy. They start to give birth to their infants in spring. And then lactation lasts from 5 to 8 months. And the lactation period can be characterized as an early period. There's peak lactation, which is when infants are under maximal velocity of growth and are very behaviorally active, but are not really starting to exploit solid food. And then you have this weaning period, which can be variable between 5 to 8 months in our population. And the reason that it's variable is because mothers need to be in positive energy balance in order to conceive at the next breeding season. Okay? Because if they miss that breeding season, they don't get another chance to reproduce for a whole year. Okay? And this is what I mean when I say that natural selection has shaped suites of adaptive traits in mothers to maximize their lifetime reproductive fitness, not just a one-shot interaction with each infant. And we see that the ability to synthesize milk across lactation varies as a function of where a mother's at in her reproductive career. So if you look here at this early time point, this is the first time that young mothers' mammary glands have ever lactated. They don't start out super great. And during early lactation, they're producing significantly less milk. Okay? But the composition isn't any different. Okay? So the total calories they're providing to their infant is significantly lower during this early period. By the time you get to peak lactation, maximal velocity of infant growth, these young mothers have actually caught up. Okay? And there's no difference in the volume of milk they're producing or the composition. But what's quite interesting is you see here the prime and aged mothers start out doing well. They increase as the baby gets bigger and it's more behaviorally active, requires more energy to do that. But the question then becomes, how are they able to sustain that? And what you find is that aged mothers and young mothers both significantly reduce their milk synthesis following peak lactation as you head into the breeding season. Whereas these prime age robust mothers are able to continue sustaining a high level of milk synthesis. And this suggests, and we're following up with data right now to find out if this is true, that these very young and very old mothers have to make a bigger trade-off between current and next reproduction. Whereas prime age mothers are able to continue investing in their current offspring when we think they're conceiving their next one. We find significant differences in the milk composition for sons and daughters. Sons get significantly more calories in their milk in terms of energy density. Mothers make milk that's higher in fat and higher in protein for their sons. Okay? However, this is importantly balanced by the fact that mothers make more milk for their daughters. And we're following up on this right now, so we'll have, stay tuned for behavioral data coming out next year. We suspect that what is going on is that these mothers are spending more time nursing their daughters. Okay? And the more milk you mobilize, the necessarily less rich it is in calories. Okay? And it may be this more time spent nursing that helps underpin that incredibly crucial lifelong mother-daughter bond. We're finding out about that as we speak. And we've included a number of important covariates in the model. So that's a long setup to get to what I came here to talk about, which is behavioral phenotype. Now, what does behavioral phenotype mean? For the purposes of this talk, I am going to define behavioral phenotype as the observable behavioral characteristics of an individual resulting from the interaction of its genotype with the environment. Okay, so it's really just the definition of phenotype with behavioral added. Okay? And this is often manifested in a suite of co-varying behaviors. And when an individual's behavior and/or response to stimuli are consistent across time and situation, it is considered a stable, trait-like quality of that individual. And there's a number of terms for this: personality, temperament, coping style, behavioral syndrome. And these different terms are largely overlapping, though not exactly, and they often reflect what discipline you are working in. So if you are in animal behavior, you are likely to call it a behavioral syndrome. If you are in psychology, working with humans, you're likely to call it personality. If you are in psychology, working with animals, you are likely to call it temperament. The important take-home message is that all of these terms are largely tapping into a particular dimension within an individual's behavior, and we're going to just subsume all of it easily under behavioral phenotype, okay? Now, one of the reasons that personality has not been a major focus of animal researchers is because of that word person embedded in it. And people thought that it was unforgivably anthropomorphizing to ascribe personality to animals. However, we do not ascribe personality to animals. They clearly have personalities, right? And there's been a number of really wonderful, rigorous methodological techniques that have been utilized to understand primate or other animal personalities or temperaments. So we're going to skip past all the methods on that entire field because it's quite elaborate. But basically, the question then becomes, why do animals have personality? Is this just individual noise, or might personality be a specific target of selection? Okay? And why would you have personality that makes you respond quickly to a rustling bush, right? Some animals that are a nervous Nelly might hear a rustling bush, automatically assume it's a jaguar, and run away. Okay? Whereas you might have a cool hand Luke who sees a rustling bush and just assumes it must be the wind, right? Now, if you assume it's the wind and it's a jaguar, that's dangerous, and you could die. Okay? But if the bush rustles, you assume it's a jaguar, and you run away, and it was just the wind, you just burned a lot of energy. Okay? And so you can see that selection is basically working on speed of response and accuracy of response. Okay? Total accuracy requires risk of finding out, and always being responsive can be energetically costly. Okay? And so you have differential selection pressures acting in concert to produce a substantial amount of variation among individuals. Okay? And personality really affords individuals the mechanism or heuristic for quick responses, okay, that are consistent across time. So how might mother's milk influence behavioral development? I'll be talking about the biobehavioral assessment program at UC Davis, which is run by John Capitanio. So I want to give kudos to my collaborator, who's also clearly a pretty cool guy. I'm going to run through some of the aspects of this program. Since 2001, over 3,300 infants at the National Primate Research Center have undergone what is termed biobehavioral assessment. So at 3 to 4 months of age, the infants are separated from they're removed from their social group, separated from their mom, and they undergo a 25-hour assessment. Okay? During this 25 hours, they're given an intruder challenge, a video playback, their blood's drawn to look at HPA axis regulation. They undergo a series of behavioral observations and temperament ratings. And I'll be presenting data from the last two today. For the behavioral observations, the infant is brought into this totally novel environment. It's removed from its social group. This is a very, very challenging, novel situation. Okay? And we find that in this situation, some animals are able to cope and adjust to the challenge, and other animals have a much harder time adjusting to this stressful manipulation. So we look at whether or not the infants manipulate a novel object, or do they avoid it? Do they locomote and explore the environment of their new cage? Or do they spend their time crouching and clinging to the cage side? Do they eat? Do they drink? Do they do any number of these different behaviors that would suggest that adjusting or shutting down? At the end of the 25-hour assessment, a technician, it's been the same technician for the entire 11 years of the study, she rates each infant along a 7-point Likert scale for 16 trait adjectives. So things about how confident, how playful, how exploratory, how gentle, how nervous, how flexible, right? All these different things. And the important thing here is that these two things integrate both methods in behavioral animal science and psychology. One is an objective measure. What did the animal do? And the other is a subjective measure. How did the animal seem? And we find that these two, the objective and subjective measure, are incredibly highly correlated. So it lets us know that these methods are tapping into the same intrinsic qualities of that individual. So what we thought might be going on is this 1 month time point in the rhesus macaque infant is incredibly important in their development, because this is when the infants first start spending significant amounts of time off the mother's chest, nursing. Okay? So they start locomoting and playing and exploring their environment. They start doing all these things. Okay? And we thought that the milk that they get at that time point may be really, really important in shaping or calibrating their behavioral phenotype. Right? So if you get a lot of milk early in development, does that make you more playful and more exploratory? And if you get a very little amount of milk, does it make you more conservative? Right? Are you more energy conservative? You can't afford to go do those things because you aren't getting enough energy. And, sorry, I gave it away, yes. Available milk energy at this 1 month time point significantly predicted infants that were better able to adjust and cope with the novel stress of BBA at 3 to 4 months of age. Okay. And here are those data. Sorry, it's not showing up super great. Along the x-axis, we have available milk energy at 1 month of age. Along the y-axis, we have their infant activity factor, z-score, at 3.5 months of age. Okay? And we controlled for maternal age, weight, parity, rank, infant age, infant weight, and infant sex. And what we find that as the available milk energy that your mom makes increases, your ability to cope with the novel situation also increases. There's a positive relationship there. And because this is correlated with temperament, we found similarly that higher available milk energy produced infants that were more what we're calling confident, because that's a trait adjective that loads highest in the factor analysis. But it comprises traits of playfulness, exploratory, curious, things like that. Okay? And here are those data. Again, along the x-axis, we have available milk energy at 1 month and their confidence factor score later in infancy. Now, if we want to believe that it's this 1 month time point that's critically organizing this behavioral phenotype, we would want to know what the milk is like right here. So is their behavior at BBA just reflecting the milk that their mother's making at that time point? And the answer is no. It's not the milk that their mothers are making at the same time we bring them in for assessment. Rather, it's the milk during this earlier critical period of behavioral development. So we know that behavior is mediated through the brain, and there are several non-mutually exclusive things that could be going on. One is that there are specific constituents in milk, like long-chain polyunsaturated fatty acids, that are differentially building the brain, okay, that are affording this behavioral difference. It could also be that milk energy affords the behavioral experiences that then specifically influence how neurodevelopment occurs and how the brain is organized in particular areas that regulate exploration and playfulness, sociability, things like that. And it could also be things in milk that directly communicate with the infant's brain. And of course, I'm talking about cortisol, or glucocorticoids if you're talking about rodents. And it turns out that all of the other work outside of my lab, looking at constituents in milk that influence infants' behavior, have looked at glucocorticoids in milk. Okay? So I'm going to spend a little time walking you through another research program's results, and then bring it back to primates, because the most elegant work, I think, being done in glucocorticoids in milk is in a rodent model. Okay? Incredibly, incredibly elegant. Oh, but first, we know that cortisol concentrations in milk reflect the peripheral circulation of the mother. So if it's in the mother's blood, it's going to be in the milk. Within the mammary gland, glucocorticoids stimulate protein synthesis. What does it do inside the infant? Well, we know that in mammalian infants, there are glucocorticoid receptors in the stomach. Okay? And importantly, the density of those glucocorticoid receptors in the stomach decreases after weaning. And this is different than most of the other tissues in your body, because in most of your tissues, as you mature into adulthood, glucocorticoid receptor density increases. So this suggests, I think quite compellingly, that glucocorticoid receptors in mammalian infant stomachs are there to receive cortisol signals in milk. So this work out of Italy is done by the Angelucci, Catalani, Casalini team, and they've had dozens of papers that have come out since 1983. So I'm going to run through some of their highlight of their results. Okay. So they have an experimental manipulation where they provide glucocorticoids in the mother's water that she drinks ad libitum. And the concentration of glucocorticoids in the milk reflects about a mild stressor, like a quick capture, restraint, and release. Okay? So mild. And they know that when mothers ingest glucocorticoids in their milk, it increases the glucocorticoids in their milk. They suggest that it does not affect mother or the dam's behavior. Okay? But as you'll see during a lecture later this afternoon, they didn't measure licking grooming. Right? Very unfortunate. What they found is that as juveniles, rats exposed to milk corticosterone as pups, that's the analog of cortisol in primates, had better spatial memory. Okay? So their cognitive processes were enhanced. So this is a Morris water maze, which is a spatial memory task in rodents. And basically, they have to learn to find this hidden perch inside the water. Infants exposed to glucocorticoids through mother's milk learn to solve this faster, and then once they know it, they get to the perch faster, okay? Suggesting that glucocorticoids prime these infants in a beneficial way. As adults, rats exposed to milk corticosterone as pups have reduced activation of the HPA axis during a stress challenge. Okay? So here's an elevated plus maze where part of it's enclosed and part of it's open. The animals that got corticosterone via mother's milk spend more time in the dangerous open arms of the axis than they do in safer closed arms of the axis. So they show less behavioral stress and more exploration in this novel setting, and the regulation of their HPA axis demonstrates physiologically that they're less stressed during this manipulation. And as adults, males exposed to milk corticosteroid pumps have increased glucocorticoid density in their hippocampus, okay, which communicates with the hypothalamus to downregulate the HPA axis. Okay. So they think that this effect here, the reduced activation of the HPA axis and the behavioral signal of less stress, is likely being mediated in part in males through their hippocampus. What's going on in females, we still don't understand, or they still haven't told us. I imagine they're working on it right now. So what we find in a single study so far that's been reported from rhesus macaques from the BBA program by a graduate student of John Capitano's is that milk cortisol, if it's higher at the time of BBA assessment, is associated with infants being more confident, playful, exploratory, and curious. So it's tapping into that confident factor score, just like available milk energy did. Okay? But the difference here is that we only tested it at the time of BBA assessment. It's an earlier study. But this is true for sons only. Okay? So the effects of cortisol in milk in both rodents, and seemingly also in rhesus, seem to exert the greatest effect in males compared to females. And here's the data. So you can see that the females are pretty much a kind of straight line, whereas the males have a significant increase. The higher the cortisol, the higher the confidence. So cortisol programs behavior, right? Well, unfortunately, there's some problems with that interpretation. So the previous studies did not integrate cortisol with available milk energy. So these two parallel studies are looking at different attributes of mother's milk and seemingly tapping in on similar outcomes. And this is a problem because cortisol in milk is significantly correlated with milk energy, right? Because I said earlier that cortisol stimulates protein synthesis in the mammary gland, so it's associated with protein significantly. And cortisol is a lipophilic molecule, right? So it tends to be attracted to the fat. Okay? So are these studies, is it cortisol that's having a direct effect? Or is it that it's serving as a proxy for other aspects of milk synthesis? And lastly, we tend to think of cortisol as being the quote-unquote stress hormone. Okay? And this is somewhat of a misnomer because cortisol is a metabolic hormone. Okay? And how your metabolism is working during a stressful time is really important. Okay? And this is why cortisol is implicated in stress events. Okay? But it regulates digestion, energy use, and metabolic processes. Okay? So is it that cortisol in milk is doing something about how the infant is utilizing energy, how they're timing their growth trajectory? Any number of other things could be going on that have to do with metabolic processes rather than directly with the HPA axis, as though those two things were separate. Okay? So what I did is I went back and I expanded the data set. So this is unpublished data right now. We're in process of finishing up all the analyses. But what we found is that if you look at milk at 1 month, and you look at milk at 3 and 4 months, and you look at the change in the cortisol concentration. So, and here I have change, it's actually an increase. So if the change is a positive change, the concentration of cortisol is going up over lactation. This is associated with accelerated infant growth. And here's, I built these models using AIC model selection. So not everything that I initially thought was going to go in the model ended up staying in the next two graphs. But what you find is that infants grow faster if they're bigger to start with, if they have more milk at the beginning of lactation, if mothers increase the amount of milk they're making across lactation, if their mothers are bigger. Okay? And this is a big no duh. Okay? I mean, you would expect if you're getting more milk, you grow faster. And if your mom's bigger, you're growing faster because you're bigger too. Okay? This is basic. What's important is that the early cortisol does not predict their growth rate. Instead, it's the change in cortisol. So the change of the glucocorticoid signal that they're getting from the mother is seemingly associated with their growth rate. And so we suspect that these signals from the mother are really important for how the infant is organizing its growth trajectory. We also found, though, that infants that get higher cortisol in milk at 1 month, and when the cortisol increases significantly across lactation, unexpectedly, they're more nervous. And this is in conflict with the earlier results that suggested that cortisol has a beneficial effect in making infants more confident. So here are those data. Okay? The magnitude of the effect size is quite small, but we would hope it would be. I'd be really upset or worried if 2 time-point measures of milk cortisol predicted everything about an infant's behavioral phenotype, because that would be really, really bad. But what this tells us is that higher cortisol makes infants more nervous. If cortisol increases across lactation, it makes infants more nervous. And if infants are low social rank, it makes infants more nervous. Now, something I didn't tell you, but social rank can be quite important within primates. And up until this study, no maternal rank factor was ever associated with anything about the milk that mothers produce or the infant's behavioral outcome. Okay, so this is a pretty novel result within my data set. What I also can tell you is that there is a group at UC Irvine, which has not yet published their data, but they're finding the same effect, that cortisol in human mother's milk predicts infants that are more timid and nervous. Okay. Haven't published it yet. I keep waiting for them to, because it's really cool. So right now, we have information that suggests that cortisol in milk definitely matters for infant behavioral development, but that so far, there's no consensus in that outcome. Right? But we're able to start building a conceptual model of how this might be working. Okay? So we know from substantial literature in all mammals that the mother's behavior is very, very important for the infant's behavior, and that the infant's behavior feeds back into mother's behavior. Okay, so this is a bidirectional pathway. We also know, and are going to hear about this during Francis Champagne's talk later today, that mother's behavior can directly influence the development of the infant's neurobiology and HPA axis, and that through that internal physiological pathway affects infant's behavior. We know that mother's nutrition and mother's behavior interact to affect infant behavior. And now we know that the milk that mothers synthesize is going to affect energy balance, right? Because energy balance is if you're in positive energy balance, as an infant you grow. If you're in negative energy balance, things are really bad. If you're in neutral energy balance, you're staying alive, but you're not growing, and you should be because you're an infant. And we now suspect that milk directly influences neurobiology in the HPA axis through glucocorticoids and the interaction of energy and the HPA axis to produce infant behavior. And so the next phase of our research, once funded, is going to start doing neuroimaging of these infants to understand what kind of glucose uptake is happening in different parts of their brain that we know mediate exploratory or nervous behaviors or fearful behaviors, like in the amygdala. So I just spent a lot of time talking about how mother's milk seemingly programs aspects of infant behavioral development. Right? And I mentioned earlier, is this just noise? Well, what we found was infants that were behaviorally active during BBA, right, which we inferred to mean that they were coping quite well with this novel challenge. Okay? If they're doing better coping in stressful situations as infants, they initiate reproduction at significantly younger ages in adulthood. Okay? And this is important because your reproductive career, the number of offspring you have, right, as a female mammal is constrained, right? You have a minimum amount of time you have to be pregnant, and you have a minimum amount of time that you have to lactate. Right? And so you have this time constraint in the number of offspring you have. Okay? The most important thing for your lifetime reproductive output is going to be the length of your reproductive career. Right? So part of that's how long do you survive. But one other way that you can lengthen that reproductive career is by initiating it earlier. And so we find that animals that adjust better during BBA give birth at significantly younger ages relative to others. So this is coming from 400 odd animals. And this model, using AIC model selection again, controls for that animal's social rank. So we know higher ranking females tend to initiate reproduction earlier. And we know that social rank was mediating their nervous temperament. And it also controls for their growth. So this isn't just a feature of the fact that they're bigger at younger ages. Something about their internal HPA and HPO axis is likely different that allows them to initiate their reproductive career at younger ages, which we suspect would have been favored by natural selection. So in summary, what this means is that mother's milk is not just personalized nutrition or in the form of immunofactors, personalized medicine. It's actually personalized programming in many, many dimensions, from hormonal to immune factors to nutrition to even the bacteria that is in mother's milk that helps colonize an infant's gut. Right? So in every dimension, it tells us unequivocally that milk that mother is providing her infant is specific to her and her infant, and different than that mother's milk for her infant. So the takeaway is this is what we know, or rather what we think we know right now, but there's still so much more that we need to understand. And so this is the outset of what I hope will be a long research program trying to understand these mechanisms in humans, in non-human primates, and other animal models. Thank you for your attention. Thank you to my staff and funding sources and collaborators, and I welcome any of your questions.
We have several microphones here, so I'm going to distribute them. I have a question for you.
Yes.
Turn on. Your last slide or one of your last slides showed that having a better background leads to early reproduction. That's a good thing. But earlier you talked about milk in the early phase being critical. and it's the younger mothers that didn't do as well.
Right.
So there seems to be a little contradiction, or can you clarify?
Yeah, I can clarify that. So I use young as a substitute for primiparous mothers because in natural ecological conditions, primiparity and youth are almost always highly correlated. Okay? Female mammals often initiate reproduction before they're done growing. Okay? And what we find is that the primiparous effects on milk synthesis are exaggerated in humans and in primates when the mother's particularly young, but it's always the first time. Whenever you are a primiparous mother, whether as a human, whether you're 15 or 35, your ability to synthesize milk that first time you activate your mammary gland to do so is compromised. So it's that, from an evolutionary perspective, we can think of these things as always being in tandem, but even when you decouple them, there's always a cost of being primiparous. The ability of the mammary gland to synthesize milk increases monotonically with each pregnancy. Okay? And so if you initiate your reproductive career at a younger age, you may have some negative consequences for that first infant. Okay? But you're starting your reproductive career earlier, and the next time you go to reproduce, your mammary gland is going to be better at synthesizing milk. Okay? And so it doesn't have to be perfect all the time, just more than not, for natural selection to give it the thumbs up.
Similarly, the first-time mother's womb is also less rich, and so the offspring has more of a survival phenotype as after birth, often because of that. If they're a teen mother or a first-time mom.
Yes, and you know there is debate in the fetal programming literature about the extent to which that fetal programming is shaping infant physiology to maximize their lifetime energy balance. And then there's other people that think that what it does is it's programming the infant for the resources available in milk postnatally, right? Because you think about something like a rodent, right? When it's being gestated, the signals that its mom's giving it are going to be highly predictive of what's going to be like when it's an adult, because that's happening in 3 months. Okay? We're talking about a primate. Like, the conditions during gestation are 10, 15, 20 years before when you're going to have to be an adult. And so how useful are those predictions for adulthood? I think the more compelling argument is that the signals that you're getting about the uterus, the uterine signals you're getting, are going to tell you a lot about the milk you're going to be getting postnatally. And so it seems to be calibrating for that postnatal survival rather than calibrating for something many, many years in the future. That's the argument I find most compelling within fetal programming. But it's still debated, and I think there's a good reason for it. So, yeah.
So does this work the same way in humans, that, if it's the first time you're lactating, that the volume is less? And so is there anything, since the more you nurse, the more milk you have, more or less, is this something that you should try to take into account and nurse more if it's the first time you've had a baby? Or?
So what they find is that the capacity of the mammary gland, there seems to be 2 major programming time points. Okay? So the first is hormonal signals from the fetus during pregnancy. So the function of the mammary gland actually is first calibrated during pregnancy. It can be reset postnatally within that first 5 days of lactation. Right? So if infant demand, or if you use a pump, or you basically signal to your mammary gland that demand is high, mothers will upregulate their ability to synthesize milk. Okay? If you miss that window, they plateau out at a sub-ideal amount of milk. Okay? And so there's lots of really good data on this coming from things like the NICU, the Neonatal Intensive Care Unit, and mothers that have traumatic births, which we'll be hearing about tonight in great detail. Traumatic births are associated with a number of challenges for breastfeeding and having the milk come in. And so there are mechanisms where you can upregulate the mammary gland's ability to synthesize milk. But again, we don't know a lot about this yet, and I think there's many, many areas that we need to know more. Oh. Hi.
I'm really, really grateful. Can you hear me?
Yes, yes.
I'm really, really grateful that you're doing this kind of research. I'm not a researcher, I'm more a practitioner.
Mm-hmm.
And I was just last
Yeah, I bet.
The call for research was so prevalent. It must have been 22 times in the 4 days of training that they said we need more research in
Yeah.
this area,
Yeah.
that area. There's so like your iceberg was perfect.
Yeah.
Remind us there's so much more. But there's also questions of who's generating the research and who's paying for the research.
Mhm.
There's Marianne Thompson said she had the unique and good fortune to see a video that was research paid for by Medaluba breast pump
Yeah.
company,
Yes.
and it was ultrasounds of nursing mothers.
Yes, this is from Peter Hartman's group in Australia.
Yes.
Yeah, okay.
And
Donnie Geddes.
unfortunately, it's not for our common consumption, because what they learned was that there's a response, an effect between the infant, the saliva
Mhm.
of the infant and the mother's breast milk. It was not a one-way, as they described it, not a one-way
Right. No, there's,
process.
exactly.
That the saliva was informing the mother's milk, the
Yeah.
mother's memory. It was just fascinating, but they said, unfortunately, because that didn't support what Medaluba wanted, which was pumping is as good as.
Right.
Then we're not getting that in commercials or any research or in Family Fun magazine. You know, the places where most women who
Right.
are not researchers might have time to read. I don't want to put that. I mean,
No,
some
no,
moms
no.
are doing tons of research, but
It's,
a lot of
it's,
moms don't have time.
it's unquestionably an issue. And there's, I have several answers to it, and I think I'm really happy you brought this up, because I think this is a really critically important thing to discuss, which is that funding for NIH and NSF is decreasing. And for whatever reason, during their last funding mandate at NIH in the National Institute of Childhood Health and Development, they didn't have anything in their list of targeted research efforts that had to do with early nutrition, like much less breast milk, you know, like it wasn't even on their radar for reasons I have no idea about. And so it's tricky because NIH and NSF are, you know, more removed from the goals of commercial interests. Okay? In terms of research, there's greater latitude for research. But those research opportunities are, you know, the funding rate through NIH right now is below 10% of grants submitted. Okay? So this is a big issue. But at the same time, you know, you have to be able to do research as a scientist or else you don't have a job, right? And so there's an essential tension between wanting to get research out and then being handicapped by where the sources of money are available, okay? And one thing that we could do is, frankly, if mothers spent more time writing to Congress people saying, why isn't NIH funding this? Why isn't NSF funding this? You know, if it were perceived that this was something that there was really high demand for and constituents cared about milk constituents, then that might change the funding cycle, and so that you wouldn't be beholden to commercial formula makers or pumping people or things like that, right? And so like I'm very, very fortunate in that I've never had to confront that. My funding comes from NSF and NIH. But what happens when I have grad students I need to support and postdocs, and that money's not there? Right? Like, is no information better than filtered information? Maybe, in some situations. And then the second issue you brought up, which I think is really, really important, is that the lack of dialogue between scientists and end users or clinicians or, you know, that I think it's tricky because as scientists, we have such a short amount of time that we have to do our teaching and our research and our service. But I think it's really important for there to be greater emphasis on outreach and actually disseminating this information to other people. And as Jim said in his introduction, this is why I started my blog, was to create a way to make the material that we're finding from this research available to the layperson in an accessible way. But that takes a lot of time, and it's not something that my tenure and promotion committee is going to care about, unfortunately. But there's basically a lot of constraints within the system, but I think you're absolutely right. And so say that more often, more loudly to more people, and maybe we'll see the system change.
I'm going to include what you just said about mothers communicating with members of Congress as part of my graduate class. This is a really important part of your nursing career, not just yours, but everyone else.
Yeah, absolutely. Thank you.
Katie.
One directive to those who are asking questions, please give us your name and affiliation, because we want to keep the dialogue going over the next 2.5 days. All
Right. And it's all being recorded for people that aren't able to be here for this particular talk because they're teaching and stuff like that. So,
Real
Jim.
fast, Katie. Jim McKenna, anthropology. One question, and I think you made me appreciate yet again how diverse different macaque species are, because I was thinking of your 3 to 4 months when they would be assessed. In some species, those animals would go through a form of anaclitic depression by virtue of being removed from the mother, you know, while they're still having a fairly intense physical connection. I know John, your advisor or friend, knows an awful lot about this because he did most of this work, but did you find that that could have had a significant effect on these infants?
Absolutely, absolutely. And actually what we're doing right now is we're doing a full sample reanalysis not related to milk using quantitative genetic methods because it's a pedigreed population, and basically looking at all, like with the 3,000 infants, we're able to do a much more fine-grained analysis of all the things that might be contributing to this. And that's going to inform a lot about how the milk moves forward. But I think you're absolutely right that some of the infants definitely have a shutdown period. We're finishing up a 3-year study right now where we're going to have about 300 infants for whom we have milk and BBA data. And for a substantial number of them, and what we've also done is we've gone to watch them and their weaning conflict in the corrals, right? And John has another graduate student that's in the process of publishing a lot of papers about, so some infants show depression when their mothers start mating again. Some infants are not concerned at all about the fact that their mother started ignoring them and go off to play, and some have more of a shutdown. And so we'll be able to kind of figure out who those infants are we capturing them with our milk data? Are we erroneously attributing something in milk that's about something more about that state of that infant? And so you're absolutely right that we need to know a lot more. Yeah.
I think we can take a few more questions, but take your break now if you need one, because then we'll go into the next speaker, okay?
Okay. Oh, great question. The question is, so this is Franz Duval from Emory University Psychology Department, and the question was, how do you collect the milk? And this is a really good question because for a long time, a number of people wanted to look at milk, but had a really difficult time getting the milk samples from the moms, right? Because if you basically stress them out too much, they won't let down their milk. And so what we do is we have a method where we bring the mother and infant in together. We very lightly sedate the mother, enough so that we can put her in like a little jacket, and then allow her to sit with her infant in a calm, quiet room. There's several other mothers and their infants there. And basically, we prevent the infant from nursing for several hours, but they do not show any signals of behavioral agitation of being in the jacket, like they would if you separated them physically and housed them in the same room, which is another method that other people have done. So with these methods, we've always succeeded in getting a substantial quantity of milk from every mother and have not had any negative consequences in terms of the mother-infant dynamic. So that's how we do it, but we only do it at a couple time points during lactation just in case the repeated captures and manipulation were to perturb the mother-infant dynamic. So, and one of the things that we're now going to be doing and following up is, so all the infants in the colony go through BBA. Only a subset go through my milk study. So what we're going to do is we have infants that the first time they ever get taken out of the corral, it's to come to BBA. Some came in for the 1-month milk collection. And we're going to look at that subset that came in for milk collection and the ones that never got perturbed to see if that is basically a stress inoculation event or if it alters their responsivity to later BBA assessment, because this is also something that could be going on that we have planned to analyze once the study is complete. Yes? I don't know who's next. Okay. Carrie Abley from Mary Grove College in Detroit.
Karen Ebling from Mary Grove College in Detroit. I'm wondering if there are any studies or any coming studies comparing breastfed to bottle-fed monkeys or humans.
There's tons of research on breastfeeding to bottle feeding. When you say bottle feeding, do you mean bottle feeding breast milk or bottle feeding the formula?
I'm talking about, yeah.
There are studies that are looking at this, and this is really important because somebody else had brought up that there's basically kangaroo care or skin-to-skin communication from the mother to the infant. And an NIH program officer actually wrote a paper called "Mother's Milk is Not Just a Meal at the Breast," and it talked about how we needed to do better understanding that the parallel interaction of milk going from the mother to the infant and the mother-infant interactions during breastfeeding, and that these are perturbed by pumping and bottle feeding. And I know that there are human researchers that are looking into this, but I don't know that we've gotten a lot of clear data out of it that I'm aware of. It might be there. I might just be missing it. We don't have anything planned like that with the rhesus monkeys, just because it requires nursery rearing the baby monkeys, right? Because you can't teach a monkey mother to pump and then bottle feed. It's not going to happen.
I think it takes a second to...
Apes, yeah. With apes you can do this. So we had a chimpanzee at the Arnhem Zoo that had insufficient lactation for her own offspring, so several offspring died. And then at some point we had a surplus infant because the mother was not taking care of it. And we trained that female to bottle feed that infant.
Right.
So I'm not sure, with the rhesus would never work, but a chimpanzee is a tool user, so it's not difficult for them. And then
But you were feeding formula, right?
Yeah.
Okay, so I think you're talking about bottle feeding breast milk, and I think the big constraint is going to be the pumping, an adequate supply
But
of ape milk.
there are in the world now a handful of apes raised by apes on the bottle.
Yeah.
Sort of interesting.
Yeah, so that would be very, very, very interesting. So yeah. Okay, last question.
Bridget McGann, Indiana University, of South Bend. I am wondering about this stem cell, like this novel stem cell source that breast milk is recently. I didn't read the article, but could you comment on the potential implications of that, if there are any?
The potential implications of it are ginormous. So a paper just came out.
Is that a scientific term, ginormous?
Yep, ginormous, super, super ginormous. So a paper just came out for those of you that it hasn't gotten the media attention it should, but I think it's building. They found that human breast milk is a source of stem cells. And these stem cells are pluripotent, so they can actually develop into multiple cell lines. And there's some initial suggestion that in the infant, these cells seem to be migrating to the infant's thymus, which is really, really important for immune function. Okay? So this stuff, I mean, this is just, it's not even officially published yet. It's only an early view, but it suggests that from an evolutionary perspective, there's the opportunity for cellular repair to happen postnatally. And two, from a research perspective, it can allow us to start doing stem cell research in a way that doesn't upset anybody morally. So, so both from a research and from a developmental perspective, this is going to be ginormous. Okay.
Do you think there might be like potential, I don't know if it conflict of interest or something like that, like in terms of acquiring the breast milk? You know, you have milk banks, but
right.
You know, and that there would be like an increasing demand. I don't know what the supply is like with milk banks, but I know that it's you know, a lot of people say it's expensive for people who just, you know, want to get it for, I don't know if it's different, like if you are a cancer patient or something.
Yes, I think there's going to be
research.
huge ethical considerations, especially in terms of clinical applications for stem cells from milk. I think that we're 5 to 10 to 15 years away from having to make big decisions. But you know, one of the things that they're doing is they're making they're inserting human genes in like cows, so that cows produce lactoferrin in their milk, which also has a lot of bioethical considerations. But it could be that there may be a way where you don't have to rely on human mothers for these stem cells using transgenic animal models. Now, that's its own bioethical consideration, but I think that it's very important to think about, yes, where the milk is coming from, how much is this competing with milk that should be going to babies, either that mother's baby or, you know, NICU donations or things like this. And I mean, one of the big issues in, you know, the 1700s, 1800s was wet nursing, and how 90% of wet nurses' infants died because they were allocating that milk for money to other mothers' infants. And so I think there's a whole messy slew of that stuff, which makes me thankful for once that all I have to deal with are animal rights activists doing primate research like I do. So, but I think it's an important thing to be thinking about, absolutely. So, yeah, thank you.